Okay, good morning and welcome to the last half day of the summit. We thought that 2.5 days is a long time for a conference and a summit. It's just a tip, as you see in the many areas and the broad band of topics we have to discuss. And thus, we will do today, at the end of this half day, we will open a panel for collaborative research, and we do hope that all of the ideas that some of you came over to us will be presented, and we are looking forward to a collaborative joint effort to a real lesson from nature from this unique region. So I thank you, everybody, for joining us and for being so collaborative and innovative. And before starting the first session, I was asked many times about the conference tomorrow, about the healthy aging. So I sent to most of you the program. I can tell you, just as a glimpse, that tomorrow morning it will start at 9 o'clock at the Yaglom building at the Tel Aviv University, and it will end around 6, something like that, with a cocktail at the Porter School of Environmental Studies. So you are all welcome. And new ideas about the joint venture between the healthy aging and the Dead Sea are always welcome. And now we want to start the first session, and I want to invite the chair, the Dean of the Life Sciences at Tel Aviv University, Abdul Azem Salam. No, Abdul Salam Azem, I'm sorry. But it has it all. It has the peace and it has the I asked you 3 times, but I misled it. Okay, thank you.
I told you to say Abid Azem, it's enough. Good morning, everybody. I'm very glad to see all of you here in the most beautiful place on earth, I think. It's an amazing place and a nice initiative. So we have 4 talks in this session, 3 20 minutes talks and 30 minutes keynote talk. Our first speaker, Iftach Yacoby from Faculty of Life Sciences, School of Plant Sciences and Food Security, and he tries to solve the hydrogen problems on Earth, no? Please.
That for me? I haven't started yet. Not working. Good morning to all of you. Can you hear me well? So before I start, I'm hearing impaired. Karen Avraham found that something like 15 years ago by doing PCR on me. And let's start. So if I'm not answering correctly, I either idiot or deaf, but you will never know. So today I'm going to speak about how hydrogen production can be used to study and engineer photosynthesis to the extreme. This is a photo of our beautiful university during the Ponciana tree flowering. It will be around the mid-June. I invite you all to see it much better than the cherry blossom in Tokyo, in my opinion. So, agriculture has been providing our food for the last 20,000 years. Why? Because some lazy boy as myself one day came to his dad and told him, look, Daddy, I'm tired on hunting and gathering food and no mammoth left, so we better start growing something for ourselves, otherwise we will simply starve. And since then, we have been domesticating all the cereals and apples and corn, etc., 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 and we are not hunters and gatherers of food anymore. Unfortunately, regarding energy, nothing changed, and we are simply the same Flintstone family as we were before. We are still hunting and gathering our energy, which is pure stupidity. We just need to decide that we need to start to grow it and use the sun energy to produce it. So our vision is microalgae-based agriculture, meaning it can be used for simple food, vitamin, high-value product, etc., etc. And in order to do that, we need to bypass the current situation where we simply domesticate strain, meaning that we can find some crazy bug that live here or in other parts of the desert. Here you can see Bar Algae is an Israeli company that grow an algae that called Chlorella ohadi that was isolated here in the desert. This bug multiply hourly. Think about a $1 of you multiply itself each hour. So that's the most crazy rate ever recorded for an eukaryotic organism. So our goal is molecular farming, meaning that we dream about harvesting hydrogen from an engineered enzyme. And the molecule that you see here, that our president probably know much better than myself how to describe it, contain a lot of inorganic part, and this thing can produce hydrogen at magnificent rate, 9,000 hydrogen molecules per second. And all of that is embedded inside a protein, something like the same wood that you see here. The same element that produces hydrogen at the same speed is pure platinum. But you compare protein versus platinum. So you can understand yourself what is the huge amount of interest that this enzyme gets. So today I would like to convince you that hydrogen production is the most direct and accurate tool for studying and engineering photosynthetic electron transfer. Why? Because we simply measure hydrogen that immediately released from the cells upon production. If you look in the photosynthetic membrane, I'm not going to dig into too many details, but we have these two green elements that you see here. It's simply biological photovoltaics. It's entities that get irradiation and emit electrons, okay? That happens in any plant that you see around you. These electrons are transferred through network, and what you see here is the yellow line that shows how electrons are transferred in this network to do many kinds of works. As in our life, you can connect a lot of different appliances to the power plug and make them work, right? So in photosynthesis, it's the same story, same electron, and those electrons do work. So the question is, how can we control the speed, the rate of the electron flow in the photosynthetic machinery? This is one question. The second question here, that when the electron comes here to the power plug of the system, it's called ferredoxin, how we can reroute them to different pathways, okay? Because, for example, here we see a competition between one pathway and another pathway, which is hydrogen production. In reality, we have many more pathways that are fighting on the electron source. So using hydrogen, we can track this thing very, very easily, because any electron that arrives here to the power plug immediately transfers to hydrogen, so we can measure actual hydrogen production, and directly, meaning we have hydrogen molecule, meaning electron came here. The speed of hydrogen production equal to the speed that electron come to the system. So beside me, I don't think that many other groups suggest to use the hydrogen production as a tool to study basic question of photosynthesis, one, and second, as a tool for engineering, because if we do some engineering in the system and we see faster hydrogen production, it means that we indeed make the electron transfer faster. What's the meaning of that? That in one day or another, we can put different enzymes here, completely non-natural one, and use the same engineering technique that we have developed to make them work and produce high-value products, for example. Why I believe in enzyme hydrogen production or in clean hydrogen production? Why I believe that it is the energy source of the future? First of all, as long as you do it with sunlight, it's a renewable energy resource produced by sun energy. It combustion is carbon-free, emits only water. If you go to Toyota Mirai, the commercial car that is driven by hydrogen, it mass-produced since 2015, you can collect water, pure water from the exhaust and drink it. It has dense energy content, contains more energy per kilogram than gasoline, and it can produce anywhere on the planet. And it has a growing market because more and more car companies start to produce commercial hydrogen cars. What is the potential? Here we see engineered system that a friend of mine in Pennsylvania State produced, a great chemist called John Goldbeck, and they have done chemical linkage between the hydrogen producing enzyme called hydrogenase and photosystem 1. Photosystem 1 is the biological photovoltaics. Using that system, they could reach about 11.4 kilograms of hydrogen per square meter per year. What's the meaning of that? Almost $200. Why? Because 1 kilogram of hydrogen gives you 100 kilometers. And you pay 60 shekels or $17 here in Israel to fuel 10 liters to your car. So as a customer, that's the price that you will agree to pay for this amount of hydrogen. Now, let's assume that all of our dreams come true and we are able to produce hydrogen at 1/3 of this maximal rate that they suggest. And it doesn't matter how we produce the hydrogen, using photovoltaics or using biological algae. Let's assume that we can produce 4 kilograms of hydrogen per square meter per year using some method. How much do we need for the state of Israel? So in the state of Israel, we have 3 million cars, and for fueling them, we need 500,000 tons of hydrogen per year. At 4 kilograms per square meter per year, we need 135 square kilometers to produce that. That means 0.6% of the state of Israel to produce it. And frankly, we can do it today with photovoltaics and water electrolysis. It's simply too expensive, but we have the technology to do it. So the idea is to develop more and more technological alternatives that can do that. Biological, photovoltaic, doesn't matter. What are the challenges in the biological world? So we have a fighting club, a non-fair competition over electron donor, which is the ferredoxin, the power plug of the system, between two gladiators. The one is ferredoxin NADPH reductase, produces NADPH, a molecule that is crucial for carbon fixation. And the second one is hydrogen production, okay? The FNR, the protein that's responsible for that, is physically tethered to the photovoltaic, meaning it is the first in the line to accept the electron, while the hydrogenate is just floating there. This is like me competing against Max Planck director in getting EU funding, okay? That's the same, just to give you the idea, okay? So the FNR has some connection to the electron source, and it gets most of the electron, and that's what we try to overcome. In a paper that we published last year, people were sure that all this hydrogen production can never work because the enzyme is extremely sensitive to oxygen. And they have been saying that for the last 70 years. We came with this paper and showed them, okay, look, this is the amount of enzyme, active enzyme in the system, the orange bar that you see here. And this is the hydrogen production. And that is oxygen. We measure all of that using mass spectrometer that we build a little node inside the cuvette. And as you see here, the hydrogen production stops when the enzyme is completely intact, meaning something else happening. And what's happening? Stealing. Carbon fixation simply steals the electron toward carbon fixation. And we show that. When we added, so this is the hydrogen production using the natural system, but when we added an inhibitor that stopped the Calvin cycle, hydrogen was keep accumulating on and on and on, and as you see, the rate is simply steady rate of hydrogen production. This way, we were able to prove that the main reason for the fast cessation of hydrogen production in biological system is simply competition over electron. So how can we bypass this competition? As a proud Jew, I really believe in Catholic marriage, and that's the reason why we have done a fusion, genetic fusion between the power plug of the system, ferredoxin, and hydrogenase, meaning that we force the hydrogenase to be at the most closest contact with the photovoltaics, with the electron source of the system, and that way we were able to bypass the competition. And here you can see that in bioengineered microalgae, we have around 4-fold hydrogen production versus the wild-type algae. So how can we engineer the system faster? That's the question. So we were able to tackle the competition, but how can we make more electrons running into the channel? So as you can see, I remind you again, we have electron running here through a lot of network in the system. Here, on cytochrome B6F, Albert will jump now, we have something very similar in mitochondria. We have a punishment for electron, because the electron, instead of going directly next, they are doing, half of them doing another round here, a process that's called Q cycle. Why that happen? Because the system wants to pump more proton from this side to the other side to make ATP. ATP, this is the basic energetic coin of the system. So this punishment makes the electron flow much slower. And the question, how slower? Nobody knows that, but I know the real number. Because using a mutant in which this Q cycle was mutated, we didn't publish this yet, by the way, we were able to show that while the hydrogen production from cycle to cycle, light, dark cycle, go down because the Q cycle kicks in, when we do not have it, we keep a steady high rate of electron transfer, which is translated to high hydrogen production rate. Meaning that now we are not only can engineer the competition, but with additional mutation, we also can knock out the slowing, the component that slow down the electron and make the electron flow much faster, meaning supporting much faster hydrogen production. And the number is 3-fold faster. If we have 3-fold faster electron flow, meaning that maybe the algae can go faster. And indeed, that's what we see. At low light regime, here 40 microeinstein and 80 microeinstein, we see that the mutant go much, much faster, orange line, and that you see here, mutant versus wild type. Mutant versus wild type. Sorry, mutant versus wild type, mutant versus wild type. You see that the mutant is much more green, meaning that we see faster rate of growth. So we came to this conclusion only because we measure hydrogen in the first place. So the hydrogen, using the hydrogen as a tool to track and understand photosynthesis, we were able to do it. And in conclusion, in photosynthesis, we have harsh competition. Most of the electrons go towards sugar production. About only 11% go toward hydrogen production naturally. If we know how to engineer and bypass all the competition toward hydrogen production, tomorrow we can put there any other enzyme, for example, nitrogenase. And I would like to end. I would like to thank all the funding agency and my group, and thank you for your listening. I wasn't time?
Questions? Yeah.
I'm approaching to you not to hit you, just to hear you.
Okay. So you reported values per year on hydrogen?
The number that I gave you is
but
per year.
so how often do you have to replace things, and how do you have to feed the algae? And you can't just let it run for a year.
It depends. If you are using in vitro system, no, I have a speaker. They can hear me. If you use in vitro system, it meaning what materials do you use in order to stabilize it. For that, I just got 8 million shekel, and I'm working with some scientists in the School of Dental Medicine, Lee Adler, and we develop smart material that will preserve it for a longer time. But if you use algae, if I can work with algae coming from here, I can use them for a very long time. So that's the reason, because they are very tough. The idea is to keep them between 1 week to 2 weeks of production. That's what we need in order to make it viable.
What's the labor to replace it? It'll cost you energy to replace the algae, to get new ones.
Of course it costs energy, but if you do it once in 2 weeks, it's not the same cost as you do it one in a day. But the questions you ask are very optimistic, because in order to answer all of those questions, I want to see an algae strain that can produce hydrogen at a rate of 200 micromole per milligram per hour. Currently, we see 15. So only when we reach something like 200, then I will think it even relevant, to ask those questions. At the moment, it's a great scientific tool for study.
Have you tried to find in nature such a
Sorry?
to find in nature search for algae that really produce more efficient hydrogen, or?
Yes, one of the experiments that I show here, that this is something that 4 years ago we got we got 1.5 million to grow this chlorella hardy on large field. That the picture that I show you, and this algae on the field produces hydrogen, not efficiently, but the idea that it multiplies itself hourly reduce the cost dramatically, because usually normal algae multiply themselves each 8 hours. So all of these things is really important for the cost of production.
Okay. Thank you.
Thank you very much.